Hello friends and Assalamu alaikum. Welcome back to this lecture series, and in this lecture we will talk about the amplification of gene of interest using polymerase chain reaction. First of all, we will try to define what is polymerase chain reaction. So polymerase chain reaction, this polymerase chain reaction is best defined as amplification or replication in vitro. So it is best defined as the replication in vitro. So normally replication of DNA makes its copies inside the nucleus of a cell, but here in case of PCR, what we do is that we make the copies of DNA outside the nucleus, outside the cell, inside a machine which is called as PCR. The polymerase chain reaction, this was discovered by a scientist by the name of Gary Mullis in 1985. So this discovery is a Nobel Prize winning discovery. He shared his Nobel Prize in 1993 in chemistry with another scientist by the name of Michael Smith. So they got their Nobel Prize in chemistry because of discovery of this polymerase chain reaction. So what is this polymerase chain reaction used for? Polymerase chain reaction is normally used when the quantity of DNA is very little and we want to amplify that DNA to much higher quantity. We want to make several thousand copies and several billion copies of DNA molecule. So wherever the quantity of DNA is less, for example, in case of a crime scene, the sample which the evidence which we might have can be in the form of a single follicle of hair, or it can be in the form of a single strain of blood. So that blood drop or that single follicle of hair, that can the DNA from those can be amplified, and several billion copies of that DNA can be formed by making use of this very technique, polymerase chain reaction. So polymerase chain reaction is now the reaction, that type of polymerization which is done outside cells, which is done in vitro. So now, what is the principle of this polymerase chain reaction? The basic principle of polymerase chain reaction is that if a DNA molecule is heated or it is melted by applying high temperature to it, the two strands of it will get separated. Suppose this is our DNA molecule, and DNA molecule, as we know, it is double-stranded. Double-stranded DNA molecule, if this double-stranded DNA molecule, say for example, it is heated or it is melted, by applying higher temperature to it, these two strands, they will get separated. This process is called as melting of DNA or denaturation of DNA. So if we apply high temperature to this DNA molecule, say for example, if we apply a temperature of 92 to 94 degrees centigrade, the two strands of DNA, the complementary strands of DNA, they will get separated. This separation of the DNA molecules, the separation of two strands of DNA molecules is what we call as denaturation, or what is called as melting of DNA. So first, denaturation of DNA, or this is what we call as melting of DNA. Melting of DNA, after this DNA is melt, means that it is two strands have separated. Now if we are able to add polymerase and the raw materials, that is nucleotides to it, this polymerase will synthesize new strands making each separated strand as template. So what will that result in? That will result in synthesis of two molecules of DNA. So here, suppose these are our two separated strands of DNA. Now, if these two separated strands of DNA, if in these two separated strands we are able to add the raw materials, that is deoxy trinucleotide phosphates. If we are able to add to this solution, and add polymerase, DNA polymerase, and add DNA polymerase, and this DNA polymerase will synthesize new DNA strand, making complementary these strands as template strand, each strand as template strand, and synthesizing the new strand, just like this. So here a new strand will be synthesized here, and from this very strand also a new strand will be synthesized. So this will result in formation of two molecules of DNA from a single original molecule of DNA. So one molecule of DNA, after one cycle, it gave rise to two molecules of DNA. And after, if we repeat the process, again, if we do the repetition of this process, we separate these two DNA molecules, so that will result in now formation of four strands. If we add again the same we repeat the same process, we will be able to make four DNA molecules. And if we repeat this process n number of times, we will be able to get 2 raised to power n number of DNA molecules. So this is the working principle of our polymerase chain reaction. That is, if a DNA molecule is heated up to a certain temperature, that will result in melting of the DNA, that will result in separation of the two strands of DNA, and then each separated strand will act as template for synthesis of new strand of DNA, and this new strand of the DNA will therefore then result in the formation of two molecules of DNA from one molecule of DNA. So from one molecule we have obtained two molecules of DNA after first polymerase, after first cycle of reaction. If we repeat the process multiple times, n number of times, the number of molecules of the DNA which we will be able to produce will be 2 to the power n. So this is the working principle of the polymerase chain reaction. Let us now discuss about the details of the PCR technique. Now, these details of PCR technique, they have been given by Ehrlich 1989 in his book, which is titled as PCR technology. So the main requirements of this PCR, they include, number one, we should have a template, DNA template. Template is any such DNA strand from which we are able to synthesize new DNA strand. Suppose the DNA, the fragment of the DNA which we want to amplify, and once we treat this once we apply higher temperature to it, the separated DNA strands will act as the template. So first requirement of the technique of DNA PCR technology or PCR technique is that we should have templates. Second requirement of PCR technique is that we should have primers. Now what are those primers? Primers, these are short nucleotide sequences. These are oligonucleotide DNA sequences. These oligonucleotide sequences are usually only 18 to 12 bases long, and they are single-stranded. Primers, these are short DNA fragments, single-stranded DNA fragments, usually 8 to 12 base pairs long. The length of these primers, it should be neither too long, neither too short. They should not be too short so that they are unable to attach with the template DNA. Neither they should be too long that they take longer time to attach with the template. So they should have preferably a smaller length, usually in the range of 8 to 12 bases. You know that in the what is called as the DNA replication. In DNA replication, DNA polymerase will add the nucleotides only to the already present primer. Naturally, it is the RNA molecule which serves as the primer in the natural DNA replication. But in this PCR or polymerase chain reaction, the primers which we add are the DNA molecules, single-stranded DNA molecules, which are 8 to 12 bases long. What primers do is that they will provide free hydroxyl groups, and those free hydroxyl groups are the sites where from the DNA polymerase will add the new nucleotides according to the sequence which will be present in the template DNA strand. This was the second requirement of our polymerase chain reaction. The third requirement of this polymerase chain reaction, they are number third, the enzymes. Enzymes which we use here are the polymerase enzymes, and we don't use the normal polymerase enzyme which is normally present in our cells. Why? Because the normal DNA polymerase which is present in our cells, that is very heat labile. It denatures at a temperature more than 37 degrees centigrade, but the polymerase enzymes which are present in certain thermophilic or the bacteria which are found in the hot springs, their polymerases, they naturally possess such polymerases which can withstand higher temperature. They are functional even at 92 to 94 degrees centigrade. Therefore, the polymerases, the DNA polymerases which we use in polymerase chain reaction, they are, first polymerase which we use is called as Taq Polymerase. What is Taq Polymerase? Taq Polymerase is that DNA polymerase which is obtained from a bacterium which is called as Thermus aquaticus. So this is a thermophilic bacterium present in hot springs. From this bacterium, we obtain a polymerase which has been named as Taq polymerase. Apart from this Taq polymerase, there are other polymerases as well, which are used in the PCR. For example, there is one more polymerase, that is PFU polymerase. There is another enzyme from this polymerase PWO polymerase, and these polymerase PTH polymerase is one such another polymerase. So these polymerases, what they do is that they will read the template, they will read the sequences of the template DNA and then synthesize new DNA strand as per the sequences present in the template strand by adding the complementary nucleotide bases to it. So this was the third requirement of our DNA, this polymerase chain reaction. Apart from these three requirements, we also require in certain type of polymerases certain buffers and certain ions. Say for example, if we are using this Taq polymerase as our DNA polymerase, then we will require a concentration of magnesium dipositive ions. Usually there should be a specific concentration of this DNA polymerase enzyme, DNA or this magnesium dipositive ions, at which this polymerase will function at its optimum level. Next requirement of this polymerase chain reaction is, next stage in this polymerase chain reaction, the next requirement is that we should have the raw materials. The raw materials which are required, they include the dNTPs, deoxy trinucleotide phosphates. dNTPs, these deoxy trinucleotide phosphates, these are required for the formation of the new strand on the template DNA strands. So this is now the requirements of our PCR. So I will recapitulate what we require for the DNA, this PCR or polymerase chain reaction. We require template DNA, we require primers, we require polymerases, and these different types of polymerases we usually make use of Taq polymerase. Apart from Taq polymerase, we have other polymerases as well. And the number fourth requirement, that is that we should have different 4 types of deoxy trinucleotide phosphates. Now we will talk about the working mechanism of the PCR. Now each cycle, this PCR involves some 20 cycles. In PCR technique, when we do the amplification of our gene of interest using the polymerase chain reaction, we normally will use some 20 or more than 20 cycles. Now each cycle of this PCR, it involves following steps. First step of each cycle is the denaturation. So first step of PCR is denaturation. Second step of PCR technique is annealing. Third step is extension. Extension is also called as polymerization. So there are 3 steps of the polymerase chain reaction. First is denaturation. What denaturation means is that during denaturation, what we do is that we subject our DNA, the DNA molecule or the gene of interest, that fragment of the DNA that is being subjected to higher temperatures. Suppose this is the DNA molecule. We will subject to these higher temperatures, we will melt this DNA. The melting of this DNA is done at a temperature of 92 to 94 degrees centigrade. 92 to 94 degrees centigrade. When we add this 92 to 94 degree, when we keep the temperature, when we raise the temperature to 92 to 94 degrees centigrade, this will result in melting of the DNA. Melting of DNA means that the 2 strands of DNA will get separated. So this will result in separation of DNA strands. So that separated DNA molecules will be now like this. Here is one strand and this is our next strand. Second step is done at lower temperature. Now what we do is that we now will reduce the temperature, we will lower the temperature to some 72 degrees centigrade. So now the temperature is now kept at 54 degrees centigrade. So at 54 degrees centigrade, now we will add the primers. So now primers will now at this temperature at 54 degrees centigrade, primers will bind to each separated strand. Now primers now are of 2 types. One is the forward primer and another is the reverse primer. We know that the 2 strands of DNA, they are complementary, but they run in opposite directions. One of the strand runs in 5' to 3' direction, and it is complementary strand, it runs in opposite orientation, that is from 3' to 5' orientation. Primers add only at the 3' end. Therefore, this very primer which we will add here, which will bind in this strand of DNA, when new nucleotides are added, they will add in this direction, backward direction, that is from this to this. While as in this very strand, the primer will again get attached to 3' end. This 3' end is located here. So this this primer will move when the DNA polymerase adds nucleotides to it. This will add nucleotides in this direction. Therefore, this primer which moves in this direction, this is called as reverse primer. reverse primer. And this very primer, which moves in forward direction, in this direction, this is called as the forward primer. So the process of attached binding of these primers to the separated DNA strands to our template DNA, this process which takes place at a much lower temperature, this process is called as annealing. The second stage of our polymerase chain reaction. So during annealing, what we do is that annealing means binding. What we bind to our template is the primers, the short oligonucleotides to the our template DNA. We have two types of primers. One is forward primer and another is reverse primer. Third and last step of polymerase chain reaction is the extension or polymerization. Now this extension or polymerization, for this temperature is again slightly raised. Now temperature is raised not up to 92 degrees centigrade, but temperature is raised to some 72 degrees centigrade. So at this temperature, the DNA polymerase, or what DNA polymerase which we use commonly for PCR, is the Taq polymerase. This Taq polymerase will add the nucleotides according to the sequences present on each template strand. This Taq polymerase is actually DNA dependent polymerase. DNA dependent polymerase means that it will read DNA, and after reading the sequence of DNA, it will keep on adding the new nucleotides. Similarly, it will read this DNA template, this Taq polymerase, and keep on adding new nucleotide state, and so that this primer, it is extended. So this process of extending of this primer to each ends of this template DNA, this is what we call as extension or polymerization. So these are the three steps of PCR. I will recapitulate them. Denaturation. Denaturation is the melting of the DNA. This is done at 92 to 94 degrees centigrade. It means the separating two strands of DNA. After two strands of DNA have been separated, then temperature is lowered. At this temperature is kept near about 52 54 degrees centigrade. At this temperature, the primers, they will bind to each template strand at the 3' end. So these primers, they are of two types, forward primer and reverse primer. Number third step of the polymerase chain reaction is extension or polymerization. This step is carried out at a higher temperature but lower than the denaturation temperature, that is at 72 degrees centigrade. At this temperature, the Taq polymerase or the DNA polymerase, any DNA polymerase which we use, if suppose we have used it in this case Taq polymerase, this will extend this primer to each end, and this process will be called as extension or polymerization. This completes one cycle. Now we repeat the process. After this, now we will have the two DNA molecules. Now after polymerization, we will now obtain two DNA molecules. One will be like this. This is the original parent strand here. This is the parent strand, and this is the newly synthesized strand. This is the newly synthesized strand. In the next one, in this very strand, we again will be having two strands. This is the newly synthesized strand, and this is the parent strand. And this newly synthesized strand will have an orientation of 5' to 3' in this case, and the parent strand will have an orientation 3' to 5'. So from after the end of one cycle, from one molecule of DNA, we have obtained two molecules of DNA. We repeat the process. Second time, we will again increase the temperature. These two strands will get separated. These two strands will also get separated. Again, lower the temperature, annealing will take place. And from these four strands, 4 molecules of DNA will be formed. So after second cycle, 4 DNA molecules. After third cycle, we will have now 8 DNA molecules. So after nth time, we will have nth cycle, we will have 2 to the power n DNA molecules. So this is how this polymerase chain reaction is done, and I will be explaining this process in the animation that is to follow. So in this animation, we will see how PCR is carried out. From sample, we obtain the DNA molecule, and then from thermal cycler, we will be able to obtain several million copies of the DNA molecule. So suppose here is this machine. This is called as thermal cycler, and this thermal cycler, it carries out the temperature cycles. So this is the test tube in which we will place our target DNA molecule and other raw materials like primers, like deoxy nucleotide triphosphates, like the DNA polymerase. So all these ingredients are put in the test tube, and this is loaded in the thermal cycler. Now once we will on this thermal cycler, inside the thermal cycles, the reaction will be carried out. So suppose this is our now DNA molecule, and this is from this in this DNA molecule, this is the target DNA molecule which we want to amplify, and this target DNA molecule is subjected to polymerase chain reaction. In cycle 1, the first step is denaturation. This is carried at 95 degrees centigrade. This will result in melting of our DNA molecule. So two strands will be separated. This is the melting or denaturation. Then we have already added the ingredients, that is the DNA polymerase. We have added Taq Polymerase, for example, primers. These we have added the free nucleotides. These are the raw materials. And then the second step is the annealing step. In annealing step, primers will get attached to each template. Then the extension, which is carried out at 72 degrees centigrade, this polymerase will read the template and will keep on adding the new nucleotides according to the sequences present on the template strand. So as you can see, from one molecule of DNA, two DNA molecules is formed. Second cycle is repeated. Again denaturation, temperature is raised, 4 strands are formed. Again annealing, and this binding of primers to each of the strand. Again extension, and the formation of some 4 molecules of the DNA. So as you can see, these are the polymerases which are the synthesize extending the primers and resulting in formation of 4 molecules of DNA. In third cycle, we repeat the process. Again denaturation, separation of the DNA strands. Again annealing, again binding of primers to each of the separated strand, and again extension, which will result in formation of multiple copies of DNA. If now we repeat this process n number of times, we will obtain 2 raised to power n number of DNA molecules.